Hello everyone, I welcome you back to another important lecture. And for today, we will see on toward a major phenomenon, and that is the seed germination. In our previous class, we concluded that how come the pollination and the fertilization would be brought about by, and how come the seeds would be formed. And now, in context of the seed germination, what we would try to do is that we would now allow the young plant that was embryo within the seed to grow out of the seed. So for that, it is important that all of you must be very much clear about the structure of the seed. So overall, the seed has three parts in it. The outside of the seed is the seed coat, or we call this as the seed testa. Within the seed, we have this storage part which goes with the name of the endosperm or the cotyledon. And then we have a young plant within a seed, and that goes with the name of the embryo. If I were to be taking you to be understanding the diagram of this, so this is the typical seed that we have got on the outside. You see that this is the covering, that is the seed coat or the seed testa. And here within the seed, we have got this young plant that is the embryo, and it was formed after we made the zygote and multiple mitotic divisions happened, and we made this embryo out of it. Now the good thing about this embryo is that it further has two regions in it. It has plumule, which are the young leaves of the embryo, and this also has this radicle in it, which would be the young root of the embryo. This part that I'm highlighting for all of you, so this is referred to as the endosperm or the cotyledon, and this acts as a store of the sugar, the protein, and the fats. So again, is important from the paper point of view. Now, the other idea is that okay, fine, if I want to grow a new plant out of the seed, so what conditions would be required? So the conditions required for the seed germination, they include the availability of the water. There has to be the presence of oxygen, and then we require some optimum temperature or the warmth. From the diagram, I would be making you understand that what is an association between the seed germination and the availability of these three factors. Now, the situation is that this young plant or the embryo that we have got, since this needs to grow, and for the growth, what we know is that various mitotic divisions has to happen. The number of cells has to increase. A lot of cell membranes and the cytoplasm and the mitochondria, and many of the cell structures they have to be forming. So this would require that this embryo needs to be supplied with the nutrients. Okay. So the idea is that in the cotyledon, we have got the sugar stored in it, we have got the protein stored in it, we have got the lipids stored in them, but they are the complex substances, and the embryo cannot use it for its growth until or unless the embryo would be releasing some of the enzymes that would break down this stored sugar, protein, and the lipids, and then they could be used by it. And this would be done with the help of the water. So the first step in the seed germination is that the water it would enter into the seed. And when this is entering into the seed, so what it would do is that it would break the seed coat. And at the same time, this would be signaling the embryo to be releasing the enzymes out of it. So you see that once the water has entered and the seed coat has ruptured, so the oxygen gas would get an entry into the seed and ultimately it would reaching the embryo that we have got here. The other thing is that the same water it would cause the embryo to be releasing three major enzymes. Among them, what we have is the amylase, we have the protease, and then we have this lipase with us. And these enzymes, what they would do next is that the amylase would break down the stored sugar within the cotyledon into the glucose. The protease enzyme that we have got, so this would break down the stored protein into the amino acids. And the lipase enzymes which are getting excreted out of the embryo into the cotyledon. So it would break down the stored lipid into the fatty acid and cholesterol. So this is how that now when since we have made the small soluble molecules, so fundamentally they could be used up by the embryo for their growth purpose. I guess if I were to just read this point for you, that the enzymes that were released out of the embryo, they would break down the complex substances that we have in the cotyledon into the simple ones. The amylase is breaking down stored starch in the cotyledon into glucose. The protease is breaking down the stored protein into amino acids. And the lipases are breaking down the stored fat into the fatty acid and cholesterol. And now they have become the small molecules and they would be used by the embryo for the growth. At the same time, the thing is that why were we in the need of the warmth? Since all of this phenomenon is under the impact of the enzymes that are being released by the embryo, and we know enzymes can only be showing their optimum activity if we provide them with the optimum temperature. So this is how the seed germination would happen. At this point, the seed would be acting as a source, and the embryo would be acting as a sink because the nutrients they are being carried to the embryo from the cotyledon. Later, when the plant would grow and it would mature and would start to photosynthesize, then when it would be reaching the age of reproduction, then ultimately when this would be making the seed again, so the seeds would be acting as a sink and the mature or the adult plant would be acting as a source. So I hope this phenomenon of the seed germination would make sense with all of you.